Plants are a rich source of antioxidants and contribute to the prevention of oxidative stress involved in many acute and chronic diseases, such as cancer, cardiovascular disease, and neurodegenerative diseases. Plant metabolites, especially those with phenolic groups, are known to have antioxidant properties. Antioxidants with high antioxidant activity are found in flavonoids, phenolic acids, and low molecular weight tannins. The chemical structure and oxidoreduction potential of phenolic compounds contribute to their antioxidant properties. In addition to the number of OH group in molecules, their placement is also important. Antioxidants donate an electron or a hydrogen atom to radicals. In these interactions, a stable phenolic radical is formed, which is crucial for their antioxidant properties. The antioxidant activity of substances is determined in vitro using different tests, which are divided into tests of the ability to scavenge radicals, also called indirect methods, and tests of the ability to inhibit oxidation under certain conditions, also called direct methods. Determining the antioxidant activity of various substances is one of the most frequently performed tests. It's thus not surprising that there are more than 20 different methods in use today. In this practical work, you will be introduced to indirect methods, such as the DPPH method. The DPPH method is most commonly used method for evaluating the radical scavenging activity of natural products. DPPH, 1,1-diphenyl-2-picryl-hydrazine, is a relatively stable radical that serves as an oxidizing substrate and as an indicator molecule for the reaction. When the DPPH radical accepts a hydrogen atom or an electron from an antioxidant, it is converted to its reduced form. After DPPH radical solutions react with antioxidants, their purple color changes from dark purple to orange to light yellow. The antioxidant activity of the sample is determined spectrophotometrically at 517 nanometers based on the difference in absorbance between the test sample and the control sample. The result is expressed as a percentage of inhibition or determined from the calibration curve of a known antioxidant, for example, gallic acid. In our practical work, we will compare the antioxidant activity of different herbal drugs in vitro. The work is divided into two parts. In the first step, we will prepare 4 different extracts from powdered drugs using 70% ethanol and demineralized water as a solvent. The extracts will be prepared by shaking or ultrasonication in an ultrasonic water bath. We will perform a DPPH test to determine which extraction method is most effective. In the second part, we will further dilute the extract with the highest antioxidant activity, perform the DPPH test again, and evaluate the antioxidant activity relative to gallic acid, that is, GAE, gallic acid equivalent. To prepare the extracts, weigh a total of 200 mg of the powdered herbal drug into 4 plastic centrifuges. Add enough 70% ethanol or demineralized water to obtain approximately 10 ml of a solution with a concentration of 20 mg per milliliter. 2 samples are lightly shaken on a shaker for 15 minutes, while the other 2 are extracted using an ultrasonic water bath for the same length of time. After extraction, filter samples through filter paper into glass test tubes. Transfer 1 ml of the filtrate to a new test tube and add 9 ml of 70% ethanol. The samples prepared in this way will be used to determine or evaluate the antioxidant activity. First, we will compare the antioxidant activity of 4 extracts. The DPPH test is performed on microtiter plates. Add 150 ml of each extract to the wells in 3 replicates. Using a multi-channel pipette, add 150 microliters of DPPH solution at a concentration of 0.1 mg per milliliter to each well. A rapid change in the color of the solution is observed. The greater the discoloration, the more antioxidants are present in the sample. We must also apply a control sample, that is, only a solvent without a sample, and a blank sample, meaning a solvent to which no DPPH has been added. Cover the plate with a lid and aluminum foil and carefully place it on a shaker, where it is gently shaken for 30 minutes. After incubation, we will measure the absorbance of samples at 517 nanometers. Carefully insert the plate into the automatic reader and measure the absorbance. Review the results and use the equation on the screen to determine the extract with the highest DPPH inhibition, with which we will continue the practical work. Next, we will prepare serial dilutions for the extract with the highest antioxidant activity and perform the DPPH test again. Add 300 ml of the sample with the highest antioxidant activity to the first well of the microtiter plate and add 150 ml of 70% ethanol to the remaining 6 wells of the same row. Then transfer 150 ml of the sample from the first well to the next well and mix the content. Using a new pipette tip, transfer 150 ml of the mixture from the second well to the third well, mix the content and discard the pipette tip. Prepare dilutions in the remaining wells following the same procedure. Discard 150 ml of the mixture from the last well. Prepare the sample dilutions in 2 replicates. Do not forget to apply the DPPH control and blank samples on this plate as well. After all samples have been applied to the plate, add 150 ml of the DPPH solution to each well using a multi-channel pipette. A more or less pronounced color gradient from purple to orange-yellow tones will appear, depending on the type of herbal drug and concentration of antioxidants. Cover the plate with a lid and protect it from light with aluminum foil. Again, incubate the plate in the dark for 30 minutes with gentle shaking and then measure the absorbance at 517 nanometers. Based on the results obtained, we determined the dilution at which the absorption was between 0.4 and 0.6, and using the calibration curve of gallic acid, we determined the concentration of gallic acid and calculate the equivalent of gallic acid or GAE according to the equation on the screen. The GAE values obtained for the studied herbal drugs are compared and the herbal drugs with the highest antioxidant activity are identified. The DPPH method is fast, simple and inexpensive. It is suitable for determining the antioxidant activity of pure compounds, extracts or biological samples in vitro. It can also be used for thermolabile compounds. The disadvantage of the method is that DPPH is an exogenous radical that dissolves only in organic media, so the antioxidant activity identified does not reflect the potential of antioxidants in the biological system. To overcome the above limitations and obtain more biologically relevant results, we perform several different tests to capture as many antioxidants as possible in the samples and their different mechanisms of action.